Welcome everybody, we're live. We're just going to give a few minutes for people to join us this evening. Teresa and Mary and I are ready for another wonderful evening on the science of COVID-19. So give us a few minutes to get started. How are you, Teresa?
I'm doing well. How are you tonight?
Doing great. Very excited for lecture number 2.
Yes.
And how about you, Mary?
I'm doing fine. Yeah, I had a busy day in the classroom, but I'm ready for a new audience. Yeah.
Great. Excellent. Okay, so we'll give it maybe one more minute and then we'll get started. Does that sound good to you, Teresa?
Yeah, that sounds great.
Perfect. Great. You have some fans in the chat, Mary.
Yay!
Yay.
My students,
Okay.
God bless them. Yeah.
Okay, so we'll get started now. A very warm welcome to our co-hosts this evening, which it'll be Mary Ellard-Ivey primarily providing the lecture, but we're very excited also to have Drs. Seidel and Evan Eskew returning for our closing Q&A. We want to extend a special greeting to those joining us live for the first time today. We had quite a few people add between last week and today, so welcome to all of you. And also would like to give a shout out to the approximately 30 PLU students enrolled in this class for credit. By taking the course for credit, they've added to their workload this semester, as in addition to participating in these lectures, they're completing weekly assignments about the material they have learned over the course of the week. These weekly assignments, in turn, are helpful to the faculty involved in this event because they tell us if we're hitting our mark regarding our course learning goals. So welcome, everybody.
Hi, everyone. Nice to be here again. I'm Teresa Chabatari, and I am co-facilitating the course with Tamara. I just want to make one additional housekeeping note, and that is if you run into any technical difficulties, either during the live event or in trying to watch the video afterwards, please reach out to us and let us know, and we'd be happy to help troubleshoot. So tonight we are pleased to welcome Dr. Mary Ellard-Ivey, Professor of Biology, who will be offering our second lecture on the science of COVID-19. And her lecture will focus on testing and vaccine development. And as Tamara mentioned, we also want to welcome back Professors Shannon Seidel and Evan Eskew, who will be joining Mary for the Q&A at the end of the lecture. So thanks again to all of you for being here, and we'll turn things over to Mary.
Great. Thanks very much for that introduction, Teresa. And thanks so much to Shannon and Evan for getting us started last week with a really engaging talk and showing me that we have a really engaged audience. So normally I like to say it's lovely to see you, but I cannot see any of my audience. But I want you to know that I know who you are. I know you're out there. Alums and community members and students in my class and colleagues around campus, I'm so delighted you're here and that you've come to listen to me talk a little bit about the science of SARS-CoV-2. So the disciplinary lens that I'm bringing to this presentation is that of molecular biology. And molecular biologists study macromolecules, and they have a lot of tools and tricks used to study them. And in particular, I describe myself a molecular geneticist. I have a great interest in the genetic material, and in particular in the tools and tricks that we use to study it. And it's from here that my interest in the topic of testing for the virus and vaccine development for the virus come from. So I'm going to talk quite a bit about testing, and there'll be a little bit of depth, and I'm going to talk some about vaccine development also. So my first picture is of a virus, but it's not SARS-CoV-2. This particular virus is called 5X174, and there's no reason on earth you'd know anything about it. It's it's what we call a bacteriophage. It's a virus that infects E. coli. And the reason why I'm showing you a picture of this virus is that it was one of the very first genomes sequenced. And it was sequenced in 1977 by a scientist called Fred Sanger. And he was responsible for developing a sequencing technology that to this day we call Sanger sequencing, that's still used, a robust workhorse of DNA sequencing. And I want to tell you a little more about that. Fred Sanger received a Nobel Prize in 1980 for the invention of Sanger sequencing. Here he is receiving his Nobel Prize. I guess only white men were allowed sequence in those days, but luckily times have changed. And a couple of things to note about this project. The virus that he sequenced had somewhere over 5,000 nucleotides present. It was 5,000 nucleotides long. And here's the amazing part. It took Fred Sanger 4 years to complete sequencing this genome. It was one of the first, it was the first complete genome sequenced. So, in contrast, I want you to consider the situation with generating data from SARS-CoV-2. It was on the 31st of December, 2019, that the World Health Organization China County Office was informed of these cases of pneumonia of unknown cause in Wuhan. And it was 10 days later that the very first novel coronavirus genome sequence, 29,000 nucleotides, was made publicly available. And I'd like you to pay attention to the public part. It's important to note that this was immediately released to public databases, and open access to data like this is critically important for the scientific process. Immediately upon release of the sequence, scientists started to work on developing a vaccine, and immediately were able to undertake some of the analysis that you learned about from Dr. Eskew last week. So I have a geeky little habit, which is I like to trawl a database called GenBank, which is part of the National Center for Biotechnology Information. It's an extremely well-known database of sequences. On the 9th, on Monday, the 7th of September, there were 22,892 complete sequences in there. And I mean complete and annotated and all of the information included. And just before class today, there were 23,791 sequences in there. And this does not include swathes of data generated in other ways. Just one more image to just emphasize that point. This is from a global viral specific database, and it illustrates very nicely the rapid pace with which we have generated sequences from the virus. So yeah, so I wanted to share that with you because the whole pace of the field of molecular genetics is absolutely blinding. So I wonder, I know you're all here and listening, and we're paying good attention to my good friend and colleague, Dr. Seidel, last week when she described some of the biology of the virus, SARS-CoV-2. I wonder if you would answer the question in the chat to show me you're out there. Do any of you remember what type of genome that the virus has? Remember to change your chat to all panelists and attendees, and we'll be using the chat for most of our interactive pieces here. So any takers? Thank you for contributing. Come on, keep going. Great, thank you so much for that answer. Now they're speeding up. Now they're speeding up. Where's all my molecular biologists? Yay, now I know you're out there. It brings me joy. Okay, so the overwhelming consensus seems to be, JD, I'm so delighted to hear from you. The overwhelming consensus is that the genome of SARS-CoV-2 is RNA. It's a single strand of RNA. And RNA is a molecular relative of DNA. And if you thought DNA, I understand why, because pretty much all living things have a DNA genome, but this virus has an RNA genome. And it isn't, you don't necessarily need to understand, unless you're a molecular biologist, the exact chemical reason, the difference between both of these molecules. The name implies it. DNA is deoxyribonucleic acid, RNA is ribonucleic acid. There's a difference in an oxygen. But one thing that does matter that I'd like you to keep in mind is that RNA is more labile. It breaks down more easily than DNA. And that's actually relevant when we start to talk about, storing and distributing vaccines. So I'd like you to think about that. Okay, so this is a slide from last week showing us indeed it has a single-stranded. Oh, I love it. I'm getting such great, yeah, the word I used was labile, and that means that it breaks down easily. Thank you for the question. Where am I going next? Okay, so I want to introduce you to a notion in molecular biology called the central dogma of molecular biology. And it's one of the first things that all our incoming biology students learn and it's got to do with the concept that the flow of genetic information only goes in one way. DNA is the repository of genetic information. It's copied into RNA, and that RNA is like a transient message that gives the information on how to make the protein. And the proteins are the cellular machinery that give rise to pretty much all structure and function. And so this is the central dogma of molecular biologists. Here is an amazing fact about coronaviruses. And to quote my good friend Shannon, she said to me one day, she said, coronaviruses knock the central dogma on its head. And that was well put. They certainly do. Coronaviruses don't have any DNA stage in their life cycle. They only have an RNA stage in their life cycle. And it's relevant for you to know about this. It'll be clear to you, I hope, in a moment. Because when the virus infects its host, it brings with it its own machinery to copy its RNA genome because its host doesn't have that machinery. We don't have the ability to make RNA molecules using another RNA molecule as a template. So this machinery, this enzyme is called RNA-dependent RNA polymerase, RDRP. And I think it's fair to say that this one unassuming little protein really is the key to this virus's life cycle. So given this, this is actually a very good target for drug treatment for COVID-19. The reason being that because the host doesn't have this enzyme, the drug will have a specific effect on the virus. And indeed, you might have heard of Gilead's drug, remdesivir, and it targets this RNA-dependent RNA polymerase. And so a couple of other things that I want to tell you about this RNA-dependent RNA polymerase, something I learned that's really interesting. Most of the enzymes that copy nucleic acids, they make mistakes, they're error prone. But luckily for us, they have a proofreading capacity. They have a capacity to go back and fix the errors in copying. most viral RNA polymerases do not have this proofreading ability and therefore are prone to mutation. But what I learned that's interesting is that there are some exceptions, and the exceptions include viruses belonging to the Nidovirales order, to which the coronavirus genus belongs. So if you want to hear some good news, the good news is this enzyme has a proofreading capacity, and so its mutation rate isn't necessarily any higher than any other virus. And so the virus is, of course, evolving over the course of the pandemic, but it's not occurring any faster than expected compared to other viruses during the outbreak. So you have gotten a little more detail of the viral life cycle. This is a figure here from Dr. Seidel's presentation, showing us how it binds to a receptor on the host cell, a receptor called the ACE2 protein. And given now that you kind of know quite a bit about the virus, I wonder if you would wiggle your fingers in the chat for me, and to all panelists and attendees, tell me if you wanted to see if this virus is in a patient, what might you look for? What might you go searching for? So I'm going to give you a moment for that. Great, the answers are coming in. So what might we look for? So we can look for symptoms that will tell if they're ill, but what will we look for to see if the actual virus is there? And I'm getting lots of answers. Keep coming. Keep coming. Excellent. I love it. And my favorite answer is no idea. That is good news. It tells me that I have something to tell you that you might be interested in hearing. So I don't know is always a great answer. Yeah. Okay. Thanks so much. That's wonderful. We have all kinds of answers. So again, just you're not incorrect if you talked about like symptoms like a fever or increased white blood cells. Those would be symptoms of a viral infection. But I'm specifically thinking about what part of the virus would you look for to determine if a person is infected with that specific virus. Okay, so where am I going now? Let me move the chat. Okay. So here, I put this table together about what I'm going to talk about a little bit about strategies for testing for infection. And some of you in here said that you would look for, some of you said you'd look for viral RNA, and a couple of you mentioned the spike protein. And those were our great answers. And indeed, the two assays for COVID-19, the two tests, indeed look for either RNA. And if you've had a test, you have most likely had a test for RNA. And I'm going to talk a little bit about protein tests additionally. You may have heard a little about the Abbott test. These are the quick tests. So if you're willing to saddle up with me for a little bit here, I would like to chat a little bit about the molecular biology of how a couple of these tests work. And I'd like to start with our strategy for looking for RNA. And so before I talk about that, I want to introduce you to some tools that molecular biologists use all the time to look for RNA and protein. So remember, protein is the product of the RNA. So like we have the gene for the spike protein in the virus, and we also have the actual built protein on the outside of the virus. Yes, this is our class mascot right here. So I want to talk about the concept of molecular biology fishing because we do it all the time. So molecular biologists commonly use a fish and bait approach to identify particular molecules. And so what we can do is imagine that this here is a sequence. Let's say it's the sequence of the spike protein from the virus. And what we can do is, even if you know a little bit about the structure of DNA and RNA, you might know that these bases, which you're all familiar with, they have some specific, what we call base pairing rules. They're sticky to their partners. And so this fragment here, if this were a fragment of DNA, this DNA fragment here would specifically bind to this sequence. So it's like a very specific way to catch that gene is how I want you to think about it, using molecular hybridization. So the RNA test, the test that we most commonly use, uses a technique called PCR, which is an acronym for the polymerase chain reaction. And the polymerase chain reaction identifies and copies a DNA molecule very specifically using this bait strategy. Picture that red piece of nucleic acid only binding to that piece of DNA, only binding to that gene. And you might be wondering to yourself why the genome is RNA and now you're talking about copying DNA. Well, in a test tube, it is really easy to make a DNA copy of an RNA molecule. So the very first step would be to turn the RNA genome into DNA. So molecular biology students, expand the acronym for PCR, please, for an attendee. Thank you very much. So it's pretty easy for us to convert the RNA to DNA. And this technology, it's something that a lot of our biology students would do pretty regularly in the lab. They would do PCR. This is the machinery that we do that we use to use it. They're called thermocyclers. And I would like, if you would indulge me, I think I have time. I'm gonna draw on my whiteboard, and I'm only drawing lines, but I think you're gonna be able to see well enough. So I would invite you now to bring your attention to my whiteboard here, and I'm gonna pull it in so you can see it. And I'm going to move my camera so you can see it. And I'm going to sketch for you a little bit about how PCR works. And we're going to notice something about it. So a reminder, we might start with RNA. So they take that sample up your nose, right? We'd convert that to a double-stranded DNA molecule. Here's the DNA. And I want to just cartoon for you how the polymerase chain reaction works. Here's what we do. Molecular biologists do this all the time. We do things that you can't see, but we do it in teeny test tubes. So we would separate these two strands thusly, and then we would use
with heat.
We would use our bait tool to copy that strand, okay? And then the next step would be, I would separate the two strands again, and my lines represent the strands, okay? And then in the next step, I would go ahead and I would copy these strands. And then I would do it one more time, and under pressure, can she draw this accurately? I would do that. And then I'd copy each of these. So, excellent. I'd copy each of these. And notice I started with one double-stranded DNA molecule. I ended up with 2. And then I ended up with 4. And then I ended up with 8. Extra credit for how many I end up with the next time. Not really, there's no extra credit in this class or any of my classes, but yeah. So the point I want to make is that we are exponentially increasing the amount of RNA, where we're really amplifying it in an exponential fashion. So I told you were going to get a good amount of detail, so indulge me, there's a little bit more. The type of PCR that we use for these assays is a special version of PCR called real-time PCR. And real-time PCR, again, if you would look at this picture with me, maybe I'll get the fancy cursor. That didn't work, did it? Oh no, okay. That's all right. What we do is we use, oh, I thank you for the reminder. That never happens to me in class, right? Right, students? The story of my life. Excellent. So what happens here is we have a bait. Think of this as a bait. It's specific for the molecule we're after. It has a reporter on one end that spits out light. It has a quencher on the other end that has an atomic structure, so it specifically absorbs the light once it's in sufficient proximity to the reporter. But every time we copy the molecule, the reporter gets cleaved from the quencher and there's a flash of light. So what we can do is we can follow the progress of the amplification of essentially the RNA in real time. And, you know, a little plug for PLU, this picture here, we're fortunate we have one of these machines here at PLU. They're pretty expensive. They cost like kind of, I don't know, probably the same as a Tesla. And photo credit to John Froschauer, who I bumped into, and he took my picture in front of the real-time PCR machine. But it's relevant for the testing for you to know that it takes expertise to run this machine. The reagents are expensive. The little tubes that we use to do the reactions are expensive. Like for the lids of them, I'm going to hold one up here. The lids of them, they have special optical properties so they can actually transmit the light. And we do those assays probably in plates like this. This is a 98 well plate. And here's some data from the real-time PCR machine. This data here was generated by a PLU student. And on the x-axis is the number of times we went through this copying process. And on the y-axis is relative fluorescence units. That's how much light was emitted. And so this is a measure of the accumulation of the product in real time. And these samples here are a series of dilutions of the target nucleic acid. And I want you to notice this line here, CT, the threshold. The more dilute sample takes a higher number of cycles to cross this threshold line, but the more concentrated sample needs a lower number of cycles to cross this threshold. And John also took this video, and I wanted to show it with you because in a way where that threshold is, it's kind of something that we set arbitrarily. And yeah, so it's kind of interesting that we sort of set this threshold arbitrarily. So, so that is how this assay works. And the take home is that it has high sensitivity because we're exponentially increasing the amount of the target. It requires quite specialized equipment, expertise, and sample handling. And it is the gold standard. It is very specific. So when you were answering my question, a number of you talked about antibodies and antigens, and I want to talk now about assays that look for proteins or antigens. So I want to clear up it's a really common misconception about what are antibodies and what's an antibody test and what are antigens. So Antibodies are the molecules that your body produces to fight the virus. They are raised against proteins in the virus. They're produced in response to exposure to proteins in the virus, like the spike protein. And proteins that elicit an immune response like this are called antigens. They're called antigenic proteins. So in order to test for infection, we could look for the protein. We could look for the spike protein. Here it is here, if you want to see it one more time. We could actually look, now we're not looking at the genetic information, but rather we're looking at the spike protein here. I'm just going to go back for a moment, and I'd like to show you briefly how that assay works, and some of the ups and downs of that assay. So on my table here, here's a little aside. I mentioned an RNA assay using CRISPR, and I only bring that up because somebody mentioned it in the Q&A section last week. And so if you want to learn more about that, I don't actually know a whole lot about it, but it's very new and really requires highly specific expertise. But take a moment now and reflect with me a little bit about looking for the protein now. So now we're looking for the protein. Well, the spike protein, like any protein, is this really complicated three-dimensional structure. And this picture here I like because it's shown translucently to emphasize the complexity of that spatial structure. And that's why antibodies are so specific. So antibodies are what your body, the host generates in response to exposure to the antigen, and antibodies can bind these proteins. And we can take advantage of that to design a test and to devise a test that can look for the protein. So on the left-hand side of this slide, here's a cartoon of how these, they're called rapid antigen detection tests, how they work. Imagine this green ball here is the antigen. This red structure here is an antibody. Again, that very specific binding. The antibody attached to it has some kind of enzyme or reporter that we can make it basically emit a beam of light or a colorimetric compound. You don't need to remember this necessarily, but for those of you who want to know, it's called an enzyme-linked immunosorbent assay, or ELISA. And what I want you to notice about this is, did you notice that this test here, there's no amplification step. There's no step at which we're kind of building up or increasing the amount of the protein. This is the technology that we use in quick strep tests, for example, ovulation predictor tests, I believe pregnancy tests. This is the technology we use. And you may have heard about the Abbott test. Alexis Matrico wrote this article in The Atlantic that you may have seen, and then he was interviewed on NPR. I think Terry Gross interviewed him. And it was entitled, you know, the test that can be the solution to the pandemic. And he was referring to these tests here. They're amenable to what we call lateral flow diagnostics. So it's like a pea on a stick test, you know. You can put a sample of whatever cellular exudate, mucus, snot, essentially, on this area here. It is drawn up through this, this capillary membrane, and the antibodies and the reagents are immobilized on the membrane. And this is a control, and this is the test. And the point about these is these are completely amenable to point of care. They're amenable to doing, you know, at the school gate. These are things that could be done at home. However, there's a catch. And in order to show you the catch, I'd like to show you some data from a paper that was published recently in the Journal of Clinical Virology. And if you'd be so kind as to look at this table here with me, it's a comparison of the real-time PCR I described, viral culture, which I didn't mention, but let me tell you, culturing a virus takes a long time and is a lot of work, and the rapid antigen detection test, the RAD test, for the limits of detection. So if you look here, here's a series of dilutions of the virus, increasing dilutions. What I want you to notice is by real-time PCR, we can start detecting at a 10 to the minus 4 dilution. And you'll notice that as we get to more dilute samples, this number goes up. That's because that's the threshold value. It goes up because it's more dilute, so you need more cycles of PCR to actually see the sample. Here is the rapid antigen detection test, the stick test. And I want you to notice that it cannot detect 10 to the minus 4 or 10 to the minus 5. There's something like 2 orders of magnitude of difference in the sensitivity between these two tests. So there is a little comparison for you. Additionally, antibodies sometimes can misrecognize the protein to which they're supposed to bind, leading to more false negatives. But the bigger issue is they do have a lower sensitivity. And so I'm going to invite you to reflect with me now for a moment on what we need in tests. We need them to be rapid, we need them to be accurate, we need them to be sensitive, we need them to be accessible, and we need them to be commensurate with risk. We know that testing, there is a huge hole in testing and accessibility to testing. I don't think it's news to you that there is a huge racial disparity in those affected by COVID-19. I decided to show you some local data, but please be assured that this is countrywide. Blacks are affected at something like two or three times the effect. Hispanic and Latino populations, something like 3 or 4 times the extent. And thank you for the questions in the chat. Commensurate with risk means that people who are more at risk need to have more testing. Thank you for the question. I love that, much appreciated. And thank you to my students who've trained me to talk and watch questions in the chat at the same time. It might all fall apart in a minute, but there's where I'm going. Okay, so I'm going to just talk a little bit now about vaccines, probably just for 10 or 12 minutes, maybe 10 minutes. And then, but before I do that, I wonder if I could indulge in you again. If you want to help me gather some information, I have pasted a link in the chat, I hope. And if you wanted to go to this Google Slides and find a slide, move down if there's a lot of people on the slide. And I guess I'm going to ask you to just think for a minute and share with me, and I want to have access to all this information later. I want you to share with me, what are your thoughts about what features are desirable in a vaccine? And then I'm going to tell you about a few. So I think the access is only to PLU. I am so sorry. Others, please feel free to type in the chat. Okay, any thoughts? What do you want in a vaccine? Thank you for understanding. I couldn't figure that out in the last minute. Thank you for contributing in the chat. Okay. Safe for people with allergies. Let me scroll down here and see what else we're getting. Targets just the virus, right? Minimal side effects, cheap, doesn't need refrigeration. This is great. Keeps a long time, fantastic. Not too many side effects. This is great. This is working out well. Keep it coming. I want to hear more. Simple supply chain. Simple supply chain, yes. One shot versus a series, absolutely. So I'm going to ask you to keep populating my slides there while I go ahead and I want to talk just a little about vaccines, but please keep populating the slides, because after I talk about a few of the vaccine candidates, it'll be nice for us to be able to maybe look back at those slides and think about if those candidates meet those targets, meet those desirable targets. So a little about different types of different types of vaccines. We sometimes use, these are strategies you'll find in any textbook. Well, probably the first three you'll find in any textbook. Live attenuated vaccines, these are vaccines that are living but have been weakened somehow. Things like this, this includes things like the live oral polio vaccine that you got on a sugar cube. Maybe you're too young and you all had the inactivated vaccine in your arm. MMR is a live vaccine. Inactivated vaccines are ones where the virus has been killed and is completely incapable of causing disease. But somebody mentioned not needing multiple doses. These can sometimes need boosters. And these can sometimes need boosters. Live vaccines tend to produce a very, very robust response and often do not need boostering. The subunit vaccines are one, excuse me, I was having this problem earlier, my slides are moving on their own. The subunit vaccines are ones where you simply administer just the antigenic protein, the protein that's going to elicit the immune response and cause the specific synthesis of antibodies to attack the virus upon subsequent infection. I'm fascinated by what I'm seeing in the chat. I need to take it easy. Operation Warp Speed was an initiative for a partnership by the government and industry to pool their resources in development of vaccines. And I've been following the vaccine race, and it is a full-time job. There are about 190 candidate vaccines out there, including all types. And so I am going to spend my last 5 minutes talking to you about 3 vaccines that have the support of Operation Warp Speed and are in phase 3 clinical trials. Phase 3 clinical trials, remember, after phase 2, they know that the vaccine appears safe and induces the production of antibodies that neutralize the virus. Phase 3 is where we get the magic placebo-controlled. Half of the recipients don't get a vaccine, they get a fake version. And double-blind, both the recipient and the provider don't know which vaccine they have. They administer this to hundreds of people, send them out on their way, and look and see the protection. So the first one I want to talk about is because it's been in the news, and it is a front runner. And the one I want, and that is the University of Oxford and AstraZeneca's AZD1222. And you can totally forget that name, but that's the Oxford vaccine. And I want to tell you how the Oxford vaccine works. The Oxford vaccine is it's a subunit vaccine, and not surprisingly, the subunit the protein that is used in that vaccine is the coronavirus spike protein. But it's administered in, it kind of hitches a ride on something called an adenoviral vector. So it is another virus. It is another virus, but this virus is simply delivering the vaccine. And this virus, this adenovirus, it causes adenoviruses cause the common cold. The one they use is a chimpanzee virus. Humans are not a host for it. It's replication deficient. So here we have the gene, we can produce the protein, and we package up the subunit, just the spike protein, in this adenoviral vectors. Now, adenoviral vectors are, Johnson & Johnson try and claim they're tried and true, but to my knowledge, no adenovector viral vector vaccines have been used in humans. They've been tested in humans. A lot of the candidate vaccines for the HIV trial, for HIV, are adenoviral vectors, I believe. And there's one adenoviral vector virus, and it's a rabies vaccine used to immunize wild animals. So yeah, so that's the Oxford vaccine, and you're probably aware of the controversy around it. Trials have resumed, last I heard, but I was away from the news today. Who knows what I missed? I want to take a couple of minutes talking to you about the Moderna vaccine, which you may have heard of. This is a play on words. It's mode RNA. It's an RNA vaccine. So the Seattle-based company Moderna, they set a drug industry record with their RNA vaccine because they identified a vaccine candidate just 42 days after the coronavirus was sequenced. And they were able to do this because it's not that hard to make the RNA if you have the sequence. It's hard to make an antigenic protein. And so they had been working on a technology for delivering. These are really different. These are totally unprecedented. We're delivering a nucleic acid that encodes the antigen that's going to elicit their immune response. All other vaccines administer a protein. So this is fundamentally different. Their technology is that they have devised these lipid-based nanoparticles that they can deliver the vaccine in, and they also have modified some bases in here. And so one thing I want you to know about this vaccine, is that it has to be stored at 4 degrees below zero. And the take home from this vaccine is that this is a completely novel and unproven technology, and there are no RNA vaccines currently approved. Here's another RNA vaccine that I'm not going to tell you about, except to tell you it needs to be stored at -70 degrees, -70 degrees. So I'd like to wrap up and just invite you to reflect on the question of, do these vaccines meet your criteria for a successful vaccine? And with that, I'm going to wrap up. Thank you so much. And I'm happy to answer questions. And Dr. Seidel and Dr. Eskew said they will join me for the question and answer period. I invite you to ask your questions through the Q&A, please. And we'll try and get through as many as we can. Thanks, Evan. Thanks, Shannon.
Thanks for the awesome lecture. It was great to learn more about the vaccines and testing.
Yeah.
I'm excited what these questions will be.
Yeah, well, there's some great questions, and there's some questions in the chat already. -70 degrees centigrade or Fahrenheit? It's -7 degrees centigrade, but I think that's -94 degrees Fahrenheit. Ask me how I know that. I looked it up today. Just cold. Yeah.
It's impressive.
Yeah. Yeah, so we have lots of expertise here with my colleagues. And so let me see. So I'd be happy to address any questions that you have. What would you say is the biggest challenge to creating a vaccine for the coronavirus? the biggest, okay, it seems to me that the biggest challenge is going to be accessibility to the vaccine, given the candidates. It seems like all of these vaccines, and I'm not an immunologist, I'm not a virologist, but all of these vaccines, the front runners don't seem to be very, very accessible. They seem to have a lot of problems. So we are going to have a vaccine, but I think the distribution is going to be a bigger issue. And I'm going to ask Dr. Seidel if she's anything to add. Shannon, have you anything to add as a virologist? What's the biggest challenge for making a vaccine?
Yeah, so lasting immunity, I think. Well, yeah. I was reading a bit of the answer, so I didn't fully hear your response.
Oh,
But
no,
yeah, did you mention lasting immunity?
no, I didn't,
Yeah.
but good call.
Yeah,
Yeah.
I would say lasting immunity because there's some evidence that we don't necessarily produce long-lasting immunity to coronaviruses. So I would add that as something to be concerned about.
Yeah. So there's a couple more questions. I think.
Yeah, there's a question about the AZT that was in the name of the AstraZeneca vaccine. Does that have any relationship to the AZT drug from that was the first HIV drug?
Oh, yeah, no, it doesn't. It's just, I didn't notice that was potentially confusing. No, it doesn't. AZT is an antiviral used against HIV, but it's just the name. Yeah.
Yeah, that's a cool connection to me.
Yeah, but well spotted. Somebody asked if the vaccine needs to be stored at -70, how quickly does it degrade after the vial is taken out of the freezer? Well, Moderna won't tell me exactly what their modified bases are, but I think any molecular biologist will tell you that as soon as you take your RNA out of the freezer, you got to get moving on it. It's the curse of RNA molecular biology. So I would say quickly.
Yeah, Mary, I wanted to point out one. I moved a related question accidentally. I marked it done. But someone asked if there are no RNA vaccines that have currently been developed or approved, how confident can we be that they are safe? And someone else asked, even if they're safe, how confident are we that CDC or other authorities in the US would approve such a vaccine since they are new?
Yeah, do you want me to take that? I can take
Yes,
that. Yeah. Oh, sorry, it didn't get marked as answered. Where is it? How confident can we be? I mean, I have confidence in placebo-controlled, double-blind trials of drugs and vaccines in my life. I'm a scientist, and that's how I make decisions for my family and for my health. And the gold standard is placebo-controlled double-blind. So I would assume that they will be subject to the rigors of the CDC and other agencies. So yeah.
There's a question about how long stage 3 trials can take.
I don't know. Do you know?
I'm happy. I could say a few things about it. The stage 3 trials often have a target of infections, or so they basically enroll large numbers of people, tens of thousands. I think some of the vaccine candidates being tested right now are enrolling about 30,000 volunteers. But how long it takes depends on how quickly the virus is spreading in the place where the phase 3 trial is happening. So let's say if you started the phase 3 trial in New York right now, it would take a very long time to get the results because there are low levels of infection in New York. If you started the phase 3 trial 4 or 5 months ago in New York, we very quickly would have data. So it depends a lot on what's happening at the local level where that trial is occurring. So it all has to do with how quickly the virus is spreading within that population of 30,000 or so volunteers. So there, I think I heard something today that somebody, maybe the CEO or somebody important at Pfizer, said there's a 60% chance that there will be results from the phase 3 trial by the end of October. So take that with a grain of salt, but
Yeah.
yeah.
So I'll have an answer at this one. Somebody asked, will one vaccine be effective at treating the various strains of coronavirus? So it's my understanding that if we're targeting these critical proteins that are needed for the viral life cycle, like the RNA-dependent RNA polymerase and the spike protein, those do not mutate frequently in the virus. They're what we call highly conserved. So it's reasonable to expect that one vaccine is not going to become ineffective in a short period of time.
There's a question about the front runners. I'm assuming they're talking about the vaccine candidate front runners, and are they in Western countries, or do you have thoughts about, I know there's at least one Chinese vaccine.
Yeah, so scanning the list of potential vaccines, they are from all over the world, from every country. Yes, science occurs all over the world. And they cover all types of vaccines, like there are live vaccines and attenuated vaccines. A lot of those are in the early stage. But no, they're worldwide. Front runners, they just were advertised in the news as front runners, truthfully, and I chose them because they seem to be doing pretty well. Yeah. Evan, you're going to answer this question about how confident. Did you say you were going to answer that question?
I can take a stab at it in a slightly different way. Someone asked about the risk of the warp speed aspect of the vaccine. And I think I would agree with what Mary already laid out, is that we do have these standards for approving vaccines, and it's reasonable to expect that any vaccine developed for coronavirus is going to have those same standards. The bigger deal is that we still don't have a vaccine yet, right? That we have to go through this process. And as Shannon said, that process is subject to local conditions, and it takes us a while. That's why we're still in the position we are. So to kind of link it back with a little bit of last week's lecture, that's one thing that some folks are wanting to do, is to develop technology platforms to create a vaccine, for instance, that would be broadly applicable across groups of viruses. Because right now we have a very targeted approach where we need to develop a new vaccine for each thing that comes along, and that process takes quite a long time. So I don't think that there's necessarily a risk of warp speed. The problem is actually that we can't move fast enough when we have a pandemic situation like we're in right now. And people want to shift the whole vaccine development to enable even more rapid vaccine testing and development.
Mary, I think you should choose a final question.
There's a question here from a PLU alum, and she asks, advice for communicating to those who distrust in science and government in layman's terms. And people talking about, because we see a lot of people concerned they won't trust the vaccine, and we have worse, the vaccine, the virus is a hoax. And this is an issue, and this is one that's near and dear to my heart. And that's part of the reason, I guess, and a roundabout answer, it's part of the reason why I'm very invested in what we do at PLU and in education and science education in general. In terms of communicating in layman's terms, I mean, I guess it's good. The most helpful thing, I think, to communicate to non-scientists who are distrustful of the process of science is that science isn't some absolute dogma that somebody decided on Monday afternoon. Science is constantly evolving. It is constantly evolving and changing. So something that's in the literature one day can then change the next day. And something doesn't become dogma until endless decades of repeated science have shown that gravity is real. If I open this window and jump, I'm going to fall. So I don't know, that might be a strategy, is to explain that science is a way of looking at the world. It's done us good. It's why we have bridges and cell phones, and our babies don't die of diarrheal diseases in their first year of life, and that we can expect to live to our 60s and 70s to be cranky on social media about scientists and to, yeah. So that's my advice. I don't know. So is it time to wrap up?
It is. It's about 1 or 2 minutes after 7.
Questions are great. The questions.
Yes, we could go on for hours. Thank you so much.
To think about. Yeah.
Thank you, Mary, Evan, and Shannon, for laying a foundation for us related to the science of COVID-19, and for your 2 really extraordinary lectures, which have been informative, beautifully succinct, clear, and engaging. If we could do a full round of applause, if it were possible, we would do everything we could do to make it audible to you as a sign of appreciation. Thank you so much.
Yes, thank you. This has been wonderful, and the 3 of you really laid a really strong foundation for the rest of the course. So we appreciate you and the knowledge you shared with us. We also want to thank all of our participants for joining us this week, and we look forward to seeing you next week. Same time, you can use the same Zoom link. And our next session will be a panel of PLU alumni sharing their perspectives on COVID-19 from the field of emergency medicine. And the panel will be moderated by PLU professor Dr. Serja Hay. So we look forward to seeing you all then. And in the meantime, have a nice evening and keep yourselves safe out there.
Good night.
Good night.
Good night.
Thank you.